the 60s, herein lies a Nobel Prize for a fellow Anfinsen, is that in fact it's been proved that proteins, now I have to be a little bit, let me say some or most, or I'll be precise and say most water-soluble, globular, whatever that means, and I'll explain proteins, self-organize. So the first experiment this fellow Anfinsen, well, I don't know how much biology sort of to discuss here, but a fellow Anfinsen took a bovine pancreatic protein, took it out of the organism, denatured it, so took it from its folded state and put it actually in uric acid to make it look like this, to make it look like a coil, then decreased the concentration of the uric acid, and it folded again, and then he put it back in and saw that it was biologically active. This being the first proof that in fact the protein self-organized. It wasn't something in the cell that folded it. There have since then, now that we have the empirical tools that I'll discuss next, there have been many further proofs of this fact for a wide class of proteins. Okay, so proteins self-organize, and I guess, let me discuss hydrogen bonds and then move on to sort of the fundamental problem in molecular biology. So one aspect, and let me get to another board. I better hurry up too. One aspect of this dynamics, in fact, one key aspect of this dynamics is hydrogen bonds. So as you may or may not know, when you take an electronegative atom like an oxygen and you move it near another electronegative atom like a nitrogen that happens to be covalently chemically bonded to a hydrogen, these guys are both hungry for this electron and get very close and share it and hydrogen bond, because they again explain the mechanical aspect of sharing these orbitals. And that's called a hydrogen bond. And it's no accident that I used O's and H's and N's, because in fact, hydrogen bonding occurs among the O's along the backbone, and this chain goes on and then comes back, and back over here, and then an H bonded to some nitrogen could hydrogen bond with this oxygen. And this is one of the fundamental aspects this is one of the guiding principles and fundamental aspects of this protein folding. So one aspect of dynamics is the formation of H bonds. And maybe I'll just leave the picture at that. There's a very good question to ask here, is if this is all taking place in an aqueous solution, why doesn't this hydrogen, why don't we just form a hydrogen bond with a water molecule here and another hydrogen bond with the oxygen who would otherwise possibly be hydrogen bonding with him? You see, why bother having any sort of hydrogen bonding along the backbone when it could all be done with the water solution? And the beautiful and simple answer is that in scenario one, where there's a water here and a water here, there are two hydrogen bonds. In scenario two, where these guys, well, this guy and some distant guy are hydrogen bonded, the two waters are now free to hydrogen bond. So there are two hydrogen bonds also in scenario two, but this water dimer now can move. So there's greater entropy in scenario two. So one aspect of dynamics, which is a very beautiful, simple fact, is one aspect of dynamics is the formation of hydrogen bonds, which, for entropic reasons that I just explained, this very simple thing, want to form along the backbone rather than just with the ambient water molecules. Form along the backbone. Okay, and this will play a role in our model, and which is why I'm sort of emphasizing this. And hydrogen bonding, Kolia, is about a half a kilocalorie per mole. So relative to this 10 kilocalories per mole. Is that right? Yeah. Let me skip a further discussion of the energetics. I'm feeling a little pressed for time. And let me just now, I guess, just come back to this board and say there is, in fact, a fundamental problem in molecular biology already on the board. Namely, since proteins self-organize, there's a beautiful problem. You give me this word in this 19 or 20 letter alphabet, and I want to now predict the structure of the folded protein. So this is the folding problem. Folding problem, which according to Anfinson is, well, at least usually well posed. The folding problem is to predict the crystalline structure. Crystalline. Let's see, predict the crystalline from the chemical structure. Chemical structure. And maybe I should explain why this is this fancy. This ought to be a play prize or something. This is this incredible problem because here's the deal. Choose your favorite disease. As I've indicated, all organisms on this planet function by proteins. So in this disease, there's a protein. Maybe it does this. Now all we have to do is design a protein design another protein that comes in and blocks that function. We've thereby cured that, if we can figure out how to deliver it, have cured that disease, and you move on. So if you have an effective solution to this folding problem, say on the computer, you can design drugs, 1 million a minute, and in principle cure all human disease. That's why this is obviously big stakes. And I'm not how to put it, I'm not the only one to over-aggrandize this problem. This is sort of well known to be this fundamental human endeavor because it will lead to curing all disease incredibly. Okay, so there's this folding problem to predict crystalline from chemical structure, and maybe now in this little box, let me just mention that we're still not very good at this. We humans are still not very good at solving this folding problem. There are two main approaches, and then I better move on to the two main approaches. One of them is called molecular dynamics. Dynamics, which is just what it sounds like. You go off and are as brave as you could be from the computer science point of view and try and model all of the atoms in this complicated molecule and all of the interactions, the entropy, the electrical properties, the electrostatics, just everything, and try and model this multi-dimensional problem. And you can see because of the size of the problem, this is sort of hopeless to really do effectively. Anyway, this is one of the approaches called molecular dynamics. And the second method is called threading. And at the moment, let me just say that look, we've decided that the crystalline structure is determined by the chemical structure. So let's go off and do a bunch of experiments and figure out how this protein and this protein and this protein fold. Now we've got a new subject protein. We want to figure out how it folds. We can take little pieces of the chemical structure and say, well, that's going to be similar to how those chemicals fold in the known structure. You see? I mean, once we make this assumption that they self-organize, we can use the chemical to predict the crystalline structure against a database. Now there's some ad hoc business of some penalty function and how do you fit all these possible predictions together, but that's called this threading method. So the threading method is to empirically use known data. And I guess now that brings me to what is the known data. So we turn to that. So we finished over here. I've described the dynamical part of this, or at least to some extent, at least the hydrogen bonding, and now we move to the empirical. And then I'll describe the models. So let's see, the empirical. So now it's time for me to introduce some of the terminology in this business. The primary structure of a protein is just the sequence of amino acids along its backbone. So it's a word in this 20-letter alphabet. So the primary structure is